we have a unit to cover, right? And that I think is sufficient. So 9 classes including today's. So today we will finish off termination, and so translation is over, and we'll have one unit, which only consists of actually only two parts. That is prokaryotic and eukaryotic regulation. So say we have 8 more classes, and I would want you to look into any of the recordings or any of the classes so far. You please get back to me if you have any questions. I would want to take out some 2 classes for discussions, but I would want that the topics of the discussions be told ahead of time, because I don't want to have a scenario where I keep asking, do you have any questions, and nobody answers. Then that will be a waste of our time. Instead, if you have doubts, we will compile those questions and then have a productive discussion on that. And if you have any other additional questions, you can ask me during that class. So that would be the most productive way of utilizing the time that I'm allotting. And during the same classes, I would also want to talk about the types of questions that you might for the end sem. Okay, and some orientation will be given to you on those lines. Right now, I think you have done very well in the assignments. Majority of you have scored. You got good marks. For the last assignment, which is carrying for 4 marks, I am going to be I'm going to evaluate and try to have a critique on whether you deserve the 4 marks or not. So don't expect that I would be in the other assignments. I've been very encouraging because it takes a lot of work, preparation, and some people have done extremely well. That's fantastic. Others may not have, but yet I have tried to be encouraging and hope that you will keep improving, and many have improved, and I'm still proud of you all on that. But for this assignment, I'm going to be strict. I should be able to see that you have worked on the assignment, you understood the concepts, and you have presented it well. Okay, so please note that the marks are not going to be just like that. You have to earn your marks. Okay, keep that in mind. Yes, and I'm sure, I think some of the assignments I've seen, they're done very good job so far. So I have because we have we have the regulation part, regulation lesson, prokaryotes and eukaryotes. So I have uploaded the last year's lectures, say on prokaryotic regulation and eukaryotic. It's about I think it's 5 lectures on so we should be able to, we will cover and finish everything within these the remaining, say, 6 hours we have on for regulation. But during that, what I hope, or I'm expecting, that is why I sent those videos, is the hope that you will watch those videos and try to come for class. Okay, I'm not enforcing that you should, but you may watch them and they may be useful to you, and it will become, if you're listening to the same thing twice, it will become much more easier and better. And please also bear in mind that the videos that I've given were from the last year. It's a year back. So, and we are all trying to get used to the new technology and all those things. So there might be glitches. I might have misspoken. Just be, in any case, in the videos or anything, whatever happens, whatever is in the textbook is the final. Okay, even if now I might have misspoken or distracted or something, there might be a mistake in my video, and if you have a contention about it, what's in the textbook is final. That's how we will take it for the moment. Okay, yeah. So I hope I have given you the instruction. Yes, please.
Sir, I have a small request, sir.
Sure.
Sir, can you give us like some written assessment, like a model test for the semester exams, so it
yes,
be easier for us to
Sure,
Uh,
sure. And I will give you a model exam paper.
okay, sir. Thank you so
Is
much,
that
sir.
fine?
Yes, sir.
Yeah, please remind me if I didn't do that. But model exam paper is some of them will be straightforward, some of them will be analytical. Say, I'm talking about the assignment, right? And when I said that assignment, you will have this 4 mark assignment, one question will be out of it. So that is actually a practice, direct practice for your exam itself. So one of the questions is you'll have to do sequence analysis. And in that question, I will give you question one, and you have, okay, you all know the pattern of the exam, right? So you'll have question one, A or B, like that. Question two, A or B, like that. You have to answer 4 questions, each is for 20 marks. Okay, if you have already received the instructions, or there might be changes, but to my understanding, you will have 4 questions. Each question is 20 marks, and in each question you have A and B, and you have to answer either A or B. So you have choice within the question. So for the sequence question, you will, that is, say for example, it is the first question, you will be given sequence 1 to analyze, or you will be given sequence 2 to analyze. So that means you have to be thorough with your the compulsory assignment, because it's going to be 20 marks, and I'm already telling you that I will give you it as an exam, and you should be able to calculate those things. So you are earning marks from your compulsory assignment at 4 marks, and if you already know these things, okay, then you will get full marks. But the catch here, why I'm revealing it to you, and the point is, you should know what to look for in the sequences, and how to look for it, calculate or look at it, determine the proper ways. And in the assignment I gave you, I think 3 or 2 sets of questions, sequences. There could be, I actually have 40 sets of sequences, but I only gave 2 to not bother you too much. So you have to practice not just what is um, in the exam or in the assignment, but also many more sequence varieties could be there. So you should be able to develop a keen eye on trying to find out the nature of differences between the given sequences. So here will be a sequence set 1, and in B sequence set 2. You cannot avoid it, you have to do it, right? So 20 marks just for that. But you should be able to figure out what is the nature of change, why it happened, how it happened, how it will be repaired. It's like the making the whole essence of this course. Okay. Yeah, and I will try to make a model paper, and I will circulate, so you'll get an idea about how it is going to be. If you, I think in assignment, graded assignment 1 and 2, I made you answer CAA questions. Is that right? The old CAA papers I have given. Somebody can you confirm?
Yes, sir.
Yes. So if for graded assignment 1 and 2, I have given you old CAA questions. So the level of questions would be based on that. You're actually prepared well enough for it. Okay. But anyway, I will make one and I will circulate it before the exam. Okay. Let's get into the lesson that we were discussing, translation. And we were discussing on translation. We finished elongation, and in that process we learned a few things, and that is what I would want to discuss now. So I think just I will use this picture for review so that it will be easier to understand. Where is 1? Yeah, here is 1. We spoke about EFTU. EFTU is elongation factor TU, and it binds to GTP or GDP. And it is bound to GTP, it is active in state, and it has affinity for charged tRNA like this. This is a charged tRNA with an amino acylated tRNA. On the 3' end, an amino acid is bound. And this is the anticodon loop. And because now EFTU has affinity for it, so it will bind to that. Now you have a GTP amino acid tRNA bound EFTU. So that EFTU will come and will deliver the amino acid to the A site of the ribosome. So there are some more factors here. I mean, EFTU does not just leave away like this. Instead, it will bind here, as we have learned in the previous class. It will be bound here when the interactions, the proofreading is good. It means proofreading of the codon-anticodon pairing is fine. If it is fine, there are 3 ways of how it can be done. By the 2 adenines, 1492 and 1493, they will check if the there will be conformational changes because of the interaction. If they're fine, those 2 flip out. If they're not fine, if this interaction is not fine between the codon and anticodon, those adenines will not flip out. That would tell or kind of have a scenario where it determines the ribosome kind of senses that it's not proper base pairing between codon and anticodon. The second way is if the base pairing codon and anticodon are not fine, the EFTU, when it brings in the tRNA, charged tRNA, it doesn't fit into the factor binding site. Factor binding site is important for the stimulation of GTPase activity. So if it doesn't fit, then the tRNA along with the EFTU will be ejected out. This third way, if assume that the first 2 mechanism happens, the third way is called accommodation. Usually the amino acid is present towards this side and the tRNA moves or turns towards this side, right? That is called as accommodation. Accommodation will only happen if the codon and anticodon interactions are good. So while we think of the even though we have these many proofreading mechanisms, there are still some mistakes that happen, and that's quite normal. So that is the job of EFTU. When it brings here, it binds to the factor binding site, and when the interactions are fine, GTPase activity is stimulated, because of which the GTP is hydrolyzed to GDP, and the GDP is when it is bound to GDP, EFTU loses affinity for the tRNA and it's expelled out. And then you have the peptidyl reaction happening that happens within the peptidyl reaction center. Peptidyl reaction center, we have learned about the importance of 2' OH as well. I hope you will remember that. And one more thing we forgot is for the exchange of, for EFTU, exchange of GDP with GTP should be done. That is done by elongation factor TS. So once the peptidyl transferase reaction also has happened, we have the next step is called as elongation, which is mediated by EFG. And EFG is also a GTP binding protein. It enters into the nearby A site somewhere here, and it will kind of pushes the whole mRNA and the tRNA's 3 codons towards the site. So the ribosome is actually moving this way. You can take it either way, it's moving. So the incoming tRNA is in A site. Peptidyl transferase reaction has happened. Now it is consisting of the whole peptide here. And when it is pushed this way, now the what was in the A site, the blue tRNA is indicated here, is now in the P site, peptidyl site. And now you have an empty A site for the next round of tRNA, charged tRNA to come in. So that's about elongation. And we also learned about the next one is we learned about termination. We discussed a few things about class 1 and class 2. Class 1 termination factors or release factors, RF1, they are two classes based on recognition. Class 1 recognize the stop codons. Class 2 will mediate the termination to occur. Okay. So in prokaryotes, you have RF1 and RF2. They recognize RF1 recognize UAG and RF2 recognizes UGA. RF1 and RF2 both recognize UAA. In eukaryotes, ERF1 that itself recognizes all the three stop codons. So the job of the release or termination has three parts. That is recognition of stop codons, peptide release. We shouldn't say hydrolysis. That's wrong. If you say hydrolysis, what does it mean? You have peptide, which would mean that breaking up the whole. Instead, we should probably, there is a hydrolytic reaction, but it's only at the between the tRNA and the peptide. That is where just one hydrolysis. And eventually, we also want the ribosomes to be recycled for further initiation in the next ones, right? So first one is RF1 binds here, and it has two domains, which is called as, one of it is called as anticodon, peptide anticodon, and the other one has a GGQ. It is the motif within the, that is glycine, and glutamine. Q is for glutamine. So on one side, it is recognizing the stop codon. On the other side, it is stimulating the hydrolysis or release of the the newly synthesized peptide. Right? So release factor 1 or the class 1 release factors accomplish both the activities. And what we now have is there are two RNAs, sorry, two tRNAs occupying peptidyls P site and A site. Right now, these two tRNAs should be removed. We also want the removal of the release factor also. Only then the ribosome subunits can be further used in the next step. So that is the job of RF3, release factor 3, that is class 2 release factor. And once it binds to the release factor 1, the release factor 1 is RF1 is released out, and then again you have to remember that this is also a GTPase, but it has different activities. When it is bound to GDP, it has affinity for the release factor 1 that is bound in the ribosome, as shown in here. And once the release factor 1 is released, then there is also an exchange of the GTP. So GTP is binding and GDP is released out. And it is still occupying the same factor binding site. Remember that this factor binding site is interacted by many proteins. So far, we have at least 2, 3 or so. So, and once the GTP is bound, and the factor binding site will stimulate the GTPase activity, and then GTP is hydrolyzed to GDP, and now RF3 loses the affinity for A site or factor binding site, and it is released out. So the job of RF3 is to remove these release factors from the A site. There is a purpose for it, because we want the ribosomal subunits to be recycled to initiate translation somewhere else. This picture is from one nice review that is there. You can see that here is a stop codon. I mean, if you're trying to visualize it in a crystal structures, X-ray diffraction and so on, that is how it's likely visible. I mean, these are representations. They are not exactly how they would look like. This is probably how a ribosome would look like. And the 30S subunit is this one, and this is the 60S subunit. For our understanding, we use schematics. And the picture that I'm showing here is a three-dimensional representation of the groups. It doesn't mean that that is how the atom would look like or the molecule would look like, but they are spatially they are organized in such a way. So these are good representations of it. And here you see a ribosome. Sorry, this is a RF1, the one that is shown here, and this is supposedly the peptide anticodon, and that is interacting with the stop codon bases. And release factors also have, one of it is the peptide anticodon, serine, proline, phenylalanine. And on the other side, where we want the peptide to be released, there is a motif called as GGQ motif, that is glycine, glutamine. So that glutamine is interacting with the peptide, where the tRNA would be uh, bound to the nascent polypeptide. Okay, so that is how they are spatially organized. I'm telling you because somebody like Venki Ramakrishnan, who is an Indian born Nobel laureate, and he works a lot about translation and so on. As I said, translation is something that is very fascinating. When you are really interested in biology, there are two ways of going. I'll just say a point and then I'll move on. There is basic research or basic science, and then there is applied science. Okay, basic science, typically the point is about what is nature. It's about what, how, why, those kinds of questions are answered. How does translation happen? Why do we have two release factors? Why not just one, like in the eukaryotes? So these kinds of questions, trying to understand the nature of nature, is basic science. On the other side, applied science is more of how can we use this? How can we use, right? You have a problem and see if you can solve the problem using some scientific components or principles. So here it is more goal oriented. Here, this is more of knowledge oriented. Both are great. I'm not saying one is better and one is more or more than the other. What is important is to understand that they both are equally important. You know something, but you don't know what is useful for, then it's still not so much for human society or Earth in general. You want to find a solution, but if you don't know the nature of components that are available to us, then we cannot find solutions, friendly solutions. So both are equal, but as a student, as a scientist, or emerging scientist, I think people should start with basic science and then should eventually be able to apply those principles in real life. So what is happening in current situation is most students are directly getting in here without so much of basic science. So they both are important. There is no basic science there is no applied science without basic science. Basic science as such, unless applicable, is not so much of value. And for a successful scientist, should have started with basic science, trying to understand nature and trying to know how to deal with uncertainty when you don't know what it is and you have to find it. It's anybody's story, right? It's about how accurate your story is. And eventually, and hopefully, you will apply it for some use. Yes. So why I brought this up is just trying to look at how this structures ribosome functions, ribosome, how can you inhibit it, and so many other things will come up in core biology. So it's important that when you are reading the textbook, try to read it to completion. Even during the vacation, I hope you will be able to read it so that you will find it much more useful in your future. Well, in eukaryotic translational termination, it's also almost the same. So we have only one release factor that's called as ERF1, E for eukaryote, that performs both the functions of recognizing the stop codon and also interacting with the and also release of the peptide sequence. ERF3 binds at the same time. Okay. Um, yeah, they both, in unlike in prokaryotes, you find that ERF3 and ERF1 together bind to the empty A site where there is a stop codon. And then GTP hydrolysis, then you have the release of ERF3, and then you have the peptide release, and then ERF1 falling off, along with another factor that is involved that is called as RLI. And RLI is responsible for the release of the RF1 release factor, ERF1. Okay, so otherwise there are a few differences, but there are not. They are almost similar in nature. So finally, when we say ribosome is translation is complete or terminated, then we want the subunits free like this so that they can initiate translation again. If you remember, in initiation of replication, in translation, we started off with two subunits, small subunits and large subunit. This is the case with prokaryotes. IF3 binding here. This is how we started. Translation initiation is done by binding off the small subunit to the mRNA. So we want to get those free subunits like this, so initiation can again happen. So there are factors. There is a factor called as ribosome recycling factor, which occupies the A site, and then kind of which has similarities, structural similarities with that of EFT, tRNA, and so on. And then again, we have a factor called EFG, which is what is responsible for all of elongation steps to happen. During elongation, you have this translocation step, and that translocation step is mediated by EFG. So the same factor also binds to such site, and it will try to push it, make a translocation reaction happen, thereby releasing the two tRNAs here like this, and then eventually releasing the EFG. I mean, EFG falls off. Release factor is released out. Ribosome recycling factor is released out, and the two subunits are separated like this. Once, if IF3 doesn't bind, then these two are likely to rebind again. So because of IF3's presence, the two subunits are prevented from forming a full 70S ribosome here. So that's the role of ribosome recycling factor. So quickly, I'll try to review. Here is an A site. In the A site, there is a stop codon. Then you have the binding of RF1 or RF2 in prokaryotes. If it is eukaryotes, it's just one ERF1. And then we have the binding of RF3 along with GDP. With GDP, in that conformation, it has affinity for this RF1 or RF2. It binds here. GTP hydrolysis, and that will allow the release of RF1 or 2. And GTP hydrolysis, and then you will have the formation of GDP, and then RF3. RF3 also comes off. Now what you have is something like this, empty A site. The peptide is released away. Here is the peptide release. And that is where the ribosome recycling factor occupies that A site. And EFG takes over and makes a translocation step. And in doing so, EFG is released, ribosome recycling factor is released, and the 50S subunit dissociation from the ribosome also happens. And at that time, you will have the binding of initiation factor. And and then there is no longer this 50s subunit cannot rebind to the sorry, so it cannot form a subunit again. Okay, that's the summary of translation termination. So there is a question that I want you all to find out. And when you try to do a question like this, you will have a good review of whole translation itself. How many ATP or GTP molecules are consumed for the generation of a protein consisting of 10 amino acids? So what you should consider in each of these cases is how many ATP molecules or GTP molecules. Mostly you will see that majority of the transactions that are happening during translation are in the form of GTP. And so this probably has to do with the factor binding site, or it must be to do with the so many factors that have this GTPase binding domain or GTP binding domains. Either way, that's not, we're not asking this fundamental question of why GTP and why not ATP. These are events that happen through evolution. For our purpose at this stage, we are at the ground level of trying to learn how things are or what things are. So we'll just keep the question simple. I want you to actually calculate the total number of GTP or ATP involved in generation of the protein. You have to start with, you should assume that amino acids are already there. You have tRNAs are already there. Ribosomal subunits are already there. And every other initiation factors, all the factors are available, elongation factor, termination factors, and so on. If these are already given, and then to make a protein form, you should calculate how many ATP or GTP are involved in the binding of the initiation. Then per every amino acid to be added, you have a certain consumption of energy, GTP. You should consider EFT or EFTU, and also charging of EFTU, that is by EFTS. Charging in the sense here I meant is the exchange of GDP for GTP that is stimulated by EFTS. And also eventually in the termination, how many ATP molecules or GTP are used. So when you know these things, and when you are able to calculate these kinds of things, it will give you'll be able to remember a lot, and you'll also be able to appreciate how much energy is spent for every protein that a bacterium or human or anybody is producing. It's an expensive process. Proteins are produced at a lot of expense, right? So try to answer this question. You can take a snapshot of it and try to find out the answer for it. For simplicity, assume for prokaryotes. Okay, don't take again, eukaryotes may have certain differences with that of prokaryotes. For the question's sake, just use it as prokaryotes. So a question might come, this kind of question might come. So how many GTPs are consumed, and justify your answer will be there. That means you actually have to tell the whole of translation. In this step, these many are used by this protein or this factor and so on. So that is how a question is asked. And if I will make it very clear now, for the exam's sake, you have to know, you should be able to draw the diagrams. That's important because I don't want to read lengthy answers. There will not be any essay questions. You will at the most have to do some analytical work like this, or sequence analysis, or draw diagrams. Maybe for benefit of you all, I will tell you some of the figures or pathways that you should draw, and try to practice those, because in the exam hall you will not be able to draw them right away just like that. It's not easy. So you'll not be writing lengthy questions. Remember that. So I have a situation here. So what I have is sorry, there is mRNA, and this is where this is a start codon, AUG, and you have SD sequence. Assume it is bacterial, okay, and this is the length. This is actually a long mRNA, several codons in each case, and the mRNA is broken. That means the stop codon and others are actually in a different piece, UGA. This is the stop codon, but ribosome, this mRNA is broken here. What do you do with that, or what happens to it? Assume that ribosomes are already translating the mRNA, and I hope you remember the word polysome. When you have one mRNA loaded with several ribosomes, you call it a polysome. And this is the 5' side of the mRNA. The actual 3' side was somewhere here. One second. This is the 3' side, and here it is broken, and now you have a new 3' end of the mRNA. There is no stop codon. Then how can translation be terminated? And you should also remember that length of amino acid would be something like this, right? As more and more codons are read, you will have the new or the protein chain increasing in length. If you have if you have mRNA like this, it's very expensive and problematic for the bacterium. First of all, any protein that is being produced are useless. They might be non-functional because some parts of the codons are actually missing. Second thing is, you see there are 5 ribosomes are trapped in here. There is no way, because the A site is occupied, you cannot have termination because it's a sense codon in the A site. And because you cannot terminate, you have 5 ribosomes stuck on the mRNA like this. That's very expensive for the bacterium to do that. So firstly, there's 2 problems, right? One of it is the 5 ribosomes are stuck, which could have been used to translate some other better mRNA. The other problem is any product coming out of this mRNA is useless, right? So how to solve these kinds of things? How to dissolve or get rid of these complexes? That is where in E. coli you have it's a well conserved among the bacterial genomes, bacterial domain, that's called as tmRNA. T for transfer messenger RNA. I will not ask you this in the exam, but it's important for you to know how beautiful molecular biology and how ingenious things can come about through evolution. So it's called as transfer messenger RNA. This phenomenon is also called as translation. Do you remember the word trans-splicing? We used in when we are learning about splicing. That is splicing between 2 different RNA molecules, right? You have 2 exons in one in here and another here. If you can get both the exons here, that is called trans-splicing. You have translation. It is like translating 2 different mRNA, right? Let's see how it happens. This is called as the name of the RNA is called SSRA. It's transfer messenger RNA. And here you have a broken mRNA, 3' end. This is the ribosomes that are here are called as stalled ribosomes. And we need to get rid of it. This tmRNA has a special structure. And that structure is something like this part of it. It recognizes it resembles that of it folds into such a way that it resembles that of tRNA. So this part of it is tRNA, and here you have a small open reading frame, that is the mRNA part. So you have two parts to the to this tmRNA. One part is tRNA, and the other part is mRNA, which has a small open reading frame. Okay, and it is charged. It is even charged with alanine here. So this tRNA part is charged with alanine. So we have it occupies. So whenever there is an empty A site like this, EFTU along with the tmRNA occupies the site here, A site like this, and the EFTU will be released out. Now what happens is accommodation would have happened, and then transpeptidation reaction would happen. That is nothing but peptidyl transfer, and now the ribosome will start translating the open reading frame that is present in the tmRNA. Right. Next one is for aspargine and so on. So they will come by and they will translate until the stop codon comes. Once the stop codon comes, the termination reaction would occur, as we have learned. It's just a normal termination reaction. And this is the reading that is done based on what is present. Alanine, this first alanine is from the tmRNA alanine here. Okay. Then we have from here on till here, these are amino acids coded by the open reading frame that is present in the tmRNA. So what it does is all the proteins that I was telling you, they are the proteins that are being produced by this ribosome from the broken mRNA are going to be useless. They have to be degraded, targeted and degraded. tmRNA is a good solution for it, or translation is a good solution. Why do I say that? Is because it accomplishes three things. First one is it accomplishes the release of ribosomes. All the ribosomes that are kind of stuck or stalled on the ribosome, on the mRNA, broken mRNA, are going to be released, because now they are going to translate the open reading frame on the tmRNA, which has a stop codon, and ribosome release would happen. The second thing is degradation of the truncated protein. All the ribosomes that are ending up translating the open reading frame of the SSRA would end up having this tag. And if this tag of amino acids is present to the truncated proteins, then they are favorably bound by the proteases and degraded. So translation is actually able to add a tag which will signal that this protein is non-functional and can be or should be degraded. And cellular proteases prefer those proteins and they will degrade them. So that is the second uh, second objective of translation in a way. The third is now the mRNA that was present here is released, and it is because it is broken on the 3' side or so, and then that is also going up to be degraded. If it is not degraded, more ribosomes would try to initiate the translation from there. So translation provides a very ingenious solution to the problem, to a big problem, and that one solution would accomplish so many things, right? So that is how this is a beautiful Am I audible? Yes,
Yes, sir.
Yes, sir.
I hope. Okay. There are two, one more. I will stop with this slide. I think I have antibiotics should be discussed. Maybe. So there is something called as nonsense mediated mRNA decay, which is about, you remember after splicing, the mRNA is typically coated with exon junction complexes, right? And when the when there is, so open reading frame starts from here till here, and the ribosome would bind here and it will start on translating this way. As the ribosome moves, all these exon junction complexes would be eliminated from the mRNA. So that's the normal full mRNA. But because of any errors during transcription or so, and if there is a nonsense codon here, like in this case, you have the open reading frame starting, say, from here, initiation has happened, and here is stop codon, and so termination would have happened. That's a premature stop codon. And having these kinds of mRNA is bad because it will produce non-functional proteins, and there is a need for these kinds of mRNAs to be eliminated, and that is done by, because these exon junction complexes are that are downstream of the stop codon are still bound, and if they are bound, they would recruit proteins called as UPF1, 2, and 3, which will further recruit de-adenylating enzyme. So they will degrade all the poly A tail that is present on the 3 cap, and also decapping enzyme that will remove the 5 prime cap. As a result, what would happen? This mRNA is going to be degraded much faster. That is called as nonsense mediated mRNA decay. Nonsense is about this one, premature stop codon. All stop codons, if there is a change or mutation because of which a stop codon forms, you call it nonsense, nonsense codon, right? This is not about mutations. This is about any mistakes that may happen during the transcription. Okay, this is also with eukaryotes, what we have discussed just now. Translation is mostly associated with prokaryotes. In eukaryotes, I don't think there is a system like that. But there are other mechanisms, something like non-stop mediated decay. That means there must have been a stop codon here. So transcriptional, I'll just quickly stop, and in the next class, maybe we will discuss that. So, but I hope in the previous one, what we have seen is the difference between there is stop codon has formed. What will happen then? We need to get rid of it. So we know using the exon junction proteins, they will recruit the decapping and so on. In this case, a non-stop mediated decay. Non-stop means there must have been a stop codon, but because of some reason there is no stop codon here. Then what would happen? The ribosome would keep on moving, and it will even come to start reading the poly A tail. And poly A would code for lysine, 3 A's. That's the codon for lysine. So you'll have so many lysines attached to the end of the protein that is being synthesized, which will recruit something. At the end, you will have the termination, these proteins that are added, release factor ERF1, ERF3, along with GTP, and they will RLI1 is recruited, and then the termination would have happened. Sometimes endonuclease is recruited, because of which there is a cut made, and then exosomes will degrade these enzymes, the mRNA, in this way, so that no more translation would have happened from this particular mRNA. Like in the translation, polylysine tail in the protein is a good signal for being attached or recognized by proteases for degradation. If a protein has multiple lysines, then it is usually degraded away by proteases. Similarly, I think I will stop here, but the same concept will be there. You should try to understand what it means. No go mediated mRNA. That means ribosome has stalled. It's not going anywhere further. What happens then? Then you have a special factors, something like RF1 and so on. They occupy, and then they will the release, and then the the mRNA are degraded. So basically, you had three things: nonsense mediated, that is formation of a stop codon. What happens? You have recruitment of decapping enzyme and deadenylating enzyme because of um, the exon junction proteins. In this case, we had non-stop decay, mediated decay. If the ribosome did not stop and continued to translate poly-A tail, then you have a certain ways of degrading the mRNA and the protein that is formed. In the here, the ribosome has stalled because of some problem or some damage that may have happened in the ribosome. And in that case also, you'll have recruitment of these special proteins which will occupy the A site, and they will mediate the degradation of the mRNA. So that's the end of translation. In the next, I would want you to read the antibiotics. There will be some questions in the final exam also on antibiotics. Okay, I'll stop here. I'm running beyond your time. Okay, please message me if you have any suggestions about end semester exam and you need any kind of cooperation on that, and I will try to see what all I can do. So feel free